Welcome to the Bioenergetics Podcast with Harry Massey, where we discuss with the leading experts in energy, health, and personal growth. So I'm joined here by Professor Hemel Patel. I'm just going to let him introduce himself. So, Professor Hemel, it's great to see you again on Zoom. And why don't you just tell the audience a little bit about your background and most importantly, your transition and introduction to we call it the bioenergetic energy medicine,
Yeah,
quantum biology space.
we talk about that for an hour, I think, but good to see you, Harry. So I'm Hemal Patel. I'm a professor in the Department of Anesthesiology at the University of California, San Diego. I also serve as the Vice Chair of Research in the department, guiding research efforts for young people as they're navigating their careers. Um, about a couple years ago, I became the Chief Scientific Officer as well for Versailles Discovery, where we're developing novel products to assess mitochondrial health and energy and those sorts of things. And we may get to that in this conversation. I trained as a pharmacologist and toxicologist. So I got my PhD in 2002 from the Medical College of Wisconsin. And it was really learning about biological systems and how they work and adapt to stress. My PhD thesis was looking at the role of opioids in protecting the heart from ischemic injury. And so ischemia is really this deprivation of blood and oxygen and nutrients to an organ like the heart, which is very sensitive to that deprivation, and seeing if there's ways to make it more resilient, right? And so it was discovered that small bouts of hypoxia create this unique protective element in the heart, and it translates to every organ in the body. If you do these small bursts of hypoxia and then expose that organ to a massive stress, it's magically adapted. So one of the molecules we thought was involved in that process was an opioid, and it turns out that opioids have this dramatic protective effect in organs and do crazy, amazing things. When I started my postdoc at UCSD and developed my independent career, we really started to focus on mitochondrial biology. A lot of the endpoints of stress adaptation, a lot of the endpoints of chronic diseases like cardiovascular disease, neurological disorders, cancers, all of it comes down to mitochondrial functionality and dysfunction. And so we went into this deep dive. We had all the amazing tools and things to measure mitochondria in really, really fine detail.
Hey, can I
Um,
backtrack you for 1 minute? Because you said something fascinating. I mean, most people, when they hear the word opioid, think opioid crisis.
yeah,
So you saying there's actually like this hormetic beneficial effect of opioids?
there is. So I
Is,
mean,
is it sort of
there,
over egged the danger of them?
yeah, there. So there's this paper that was published, and I can't. It was a clinical paper, and one of the things they were commenting on was that during the opioid crisis, it was toxic in the sense that there were lots of lives that were being lost. But the flip side of this is there were these pristine organs that were being donated, and it could come from two different aspects, right? It's young people that are dying because of their opioid addiction and toxicity, but it could be the other flip side that maybe opioids that they're ingesting have this massive protective effect on organ preservation. And so this idea, right? When people exercise, exercise releases endogenous opioids, which causes this protective element. And so we could show that the heart actually is a major storage site for opioids. And so they get produced and stored there. The heart releases opioids in response to various signals. And when you give them exogenous opioids like morphine in really titrated doses, so this is not like we're making the animal high, at a low, low dose where you're engaging receptors and signaling, you actually cause a protective event. And the reason this works is it goes back to this idea of the small bouts of hypoxia that create this massive protection from a long bout of ischemia. It's all about what doesn't kill you makes you stronger, right? And so this idea that opioids are a toxic agent, so if you give them in small bursts, they actually create a memory in your organs, and your organ learns to adapt to that response, and it becomes preserved. And so this is this idea that there's a preconditioning effect that you can launch with chemical agents like opioids. And they, if we want to get esoteric, they couple to what's called these inhibitory G proteins. And inhibitory G proteins inhibit positive sort of drive signaling in systems. And so they shut things down. And so small bursts of this actually create a massive protective effect later on. And there's a whole biology and genetics around that.
Yeah, I love the field of hormesis. I remember when I was in 2020, I bought an RV and I was traveling through Montana, and there was this big sign saying radioactive mine as like a healing spa retreat. I was like, goodness, what's that? I went there, and there were all these people with euphorbia, leukemia, all sorts of cancers, etc. They'd basically been coming there for years, where they'd originally had cancer. They've been going into this, slightly, well, as hormetic, low dose radon gas radioactive mine, and being basically been healed, and they would all come back for top ups for a week a year. And yeah, I don't know, it's pretty fascinating. So we all get terrified of EMF, radiation, opioids, etc. But yeah, in low doses, they can actually
Yeah.
be
I
actually
mean,
an agent.
it's I think every ancient culture has this sort of adage, right? Anything in moderation is actually good for you. And so maybe a little bit of poison is actually not a bad thing, right?
Perfect. So yeah, maybe we'll just move on to, when did you first get introduced to the world of quantum biology and energy medicine? Because obviously you've had a completely traditional
Yeah.
university research background, and why on earth, with someone, you're sort of at the top of your career at UCSD, why on earth did you sort of dabble off into this energy medicine world?
I was raised as a Hindu, so I had that sort of in my tradition and blood, right? I mean, people say that that's where a lot of the origin of these deep meditative practices and things come from. And I explored this in college. I was a philosophy of religion major as well as biology, and I had a guru and learned to meditate and breathe and do all kinds of amazing things. Life happens, you forget all that. Never really thought that it did anything biologically. It made my stress go away, but sort of fell away from that. Went deep into the sciences, got my PhD, started working in systems and things we were just talking about. Um, and for 20 years at UCSD, the focus had been essentially that, trying to define the molecular endpoints for diseases and processes going on in cell systems, animal systems, and human systems. For about 10 years of that time, I shared an office with Toby Muller Bertram, who's an MD PhD pain anesthesiologist. And the idea was that by sharing an office between a PhD and an MD, you'd rub off on each other and you started working together on a big project and solve some major problem in research. We became really good friends, but never really worked together on any problems. And so he left the university, started his own private clinic, and this was during the rise of the opioid epidemic. And so he had to come up with alternative ways to manage pain in his clinic. And so there was a Joe Dispenza practitioner in his clinic that started playing around with meditations, and they saw some really amazing benefits. He went to a week-long event and was completely blown away by what he saw with all of the meditative practices, the healings, the huge potential that was being created by Joe in a very concerted phase. And then he started thinking about what that week long was, right? It's this perfect environment that Dr. Joe creates where you've got everyone captive for a week. They're eating similar foods, they're on the same kind of life cycle in terms of sleep wake kinds of things, and you're confined to a closed space. And so Toby thought this would be a perfect place to do research. And so he brought me sort of roaring back into this world. He remembered all the crazy biological molecular things that we do. And I think this was really the way to move that science to the next phase. I think meditation research had sort of fixated on looking at transcript changes, focused on immunology and things like that, but no deep sort of research that tried to answer the question of why and what and how can this be actioned. So we started with some pilot studies. We found some really interesting things. Joe had tried to get me to an event for quite a long time, and I saw the data, and it was just unbelievable. Some of the things we were seeing in week-long meditators. And so I came to an Orlando event, and I think that was the first time I met you as well at that event in 2021. Um,
Can I ask, were you thinking that you're just studying effects of meditation at that time, or were you thinking there was some sort of action at distance or quantum like type quantum effect?
I thought we were going to study someone resorting for a week and seeing the effects of that. When I actually went to an event, I'm like, this is not meditation, right? It looks nothing like a meditative practice. Yet all of the data we've collected over the last 3, 4 years shows that there's a massive effect on the body that's happening. And you can't explain it by normal science. There's something unique and powerful going on that we haven't been able to figure out. And it really involves
Yes.
the invisible world, right? The physics.
So I was going to say, like, a skeptic might say, well, when you're in a meditative state, you're breathing slower, your parasympathetic system is more, is more active, your sympathetic is a bit more damped down. That would naturally create a cascade of chemicals that's going to be more healing. But what would you say to that line of reasoning that it's actually something deeper?
I think there is, right? I've gotten through this work, we've gotten involved in a lot of bioenergy healing kinds of studies as well. And so, in situations where people are nowhere near each other, there is a healing benefit, and it's been around forever. Bioenergy with Reiki and other processes, there's lots of papers that have been published. The problem with these papers, and the problem we're seeing as well, is you can put your hands around something and see an effect um, on whatever you've got your hands around. But the question still is, how does that information move from your hands to that thing that you're healing? Whether it's a cell, whether it's a human, whether it's an animal, whatever it is. And that I think is the billion trillion dollar question and answer, right? We don't know what that magic, that stuff we can't define, see, taste, smell is. And this is where I think we really want to focus our energy. And this is where it gets really wonky, and it's hard to try to explain this to scientists here on campus when you're like, well, I put cells in a dish and someone sent, I mean, you can use the word pray, right? Essentially, or does a meditation around them, and they behave completely differently. And they look at you like you have 10 heads because it's unbelievable.
Out of curiosity, how do you explain it to them?
I say it's a very controlled experiment, right? We have cells that are in a different place that no one knows about. They come from the same lot of cells that are in this other space. And when you look at them as a comparator group, they look completely different. The only difference is the cells that have a response have been focused on. And so we're starting to devise some studies where we, and this is how science, I think, does things, is you have to break things to figure out how they work. And so we're trying to break that response. And so could we put the cells in a Faraday cage? Could we put them in a soundproof cage? Could we put them in a lead cage? What can we start subtracting away things that we think may be impacting this transition from the individual to the material that they're transforming? And I think eventually we'll get at that, but I think it's going to be more complex than that, right? I think there's going to be an element of physics that hasn't been described yet that's involved in this, and we don't have the tools to do it. And so I think it's going to take a lot of mental and sort of physical energy to figure out how to do this.
Yeah, I mean, there's a lot of different physics theories. I mean, Einstein originally said about spooky action at a distance, or you've got David Bohm who came up with the holographic universe, which is basically saying every single electron, molecule, etc., contains the rest of the universe, or is interconnected to the rest of the universe. And yeah, you've got all of these different sort of ideas, but in short, I mean, everything we've seen in our Nest Dash Energy for Life world, you can absolutely get action at a distance, and we've done experiments where we've actually scanned people in another, we can scan people in another country using a computer. Like we could scan someone in Australia with our computer in the USA, um, and they're not like, and it's on battery. It's not connected to the internet or anything like that, and yet we can see the results of what's going on in their body field. Or obviously, of course, with the distance healing effects, there's distance healers that do everything at a distance. Are you working with any like physicists? Because I mean, I feel, I think that's like where the next evolution of this needs to go, is combining physicists with.
Yeah, so we're making small strides in this. So one of my good friends is a physicist in Egypt, and so he worked with me here in San Diego for a number of years, and then took a position back in Egypt when the Arab Spring movement and everything was up to sort of move back to that space. And so he was just at the Orlando event this last round because someone actually experiences a week-long event, you can't describe it to them, right? You have to just come and see it and let your eyes sort of capture everything. And he was fascinated by all the stuff he saw, all the energy kinds of the healings and this, that, and the other, and going through the meditations. And my task for him was to start thinking about how can we devise tools and experiments to study what's happening. And the reason why he's sort of the next bridge is he's a biologist and a physicist, right? So I'm a pure biologist and I sort of dabble. I have books on tiny little dummy books on physics on my desk that I try to read and make sense of this. Whereas he's got an equal foot in physics and in biology. I think he gets us one step closer to thinking about how the biology engages with that physics. And then the next step ultimately in this is to get to the full-on physics that are theoretical and are building models and stuff that we can then start testing out. The ultimate goal for all of this is to have an institute on campus where we just sit around as, all kinds of diverse people and I think it's not only scientists, right? You need biologists, you need biophysicists, you need physicists. But then I think you need musicians, you need artists that paint and do other things that think way outside the box, that look at your stuff and start to think about it in a creative, eclectic kind of way. I think that's what's going to solve this problem and move it.
Have you seen that? There's a I'm trying to remember his name, but there's a YouTube clip. He's a professor of music, and yeah, he worked with another professor from the biology department, and he basically found that if he used I think it was a frequency is exactly 10 times the rate of the low frequency, they were helping to eradicate cancer and kill little, I think it was like bacterians and parasites. But as you say, that idea came from a music professor,
Yeah.
not a
And
medical
I mean,
professor.
I think musicians are probably like some of the smartest mathematicians because you have to understand math to do music. And I think they're very interesting frequency people as well because they can hear things that other people don't, right? And so I think there's an art that they have that could really inform the biological sciences. And there's so much power in music to heal and bring memories up and other things. And it's got to somehow connect with biological systems to do that. So I think we need a very sort of diverse group of people coming together to think about this larger problem.
That's exactly the sort of mission that if mission is the right word, but we're running this Zenergy conference in Denver on the 3rd to the 5th of October. But it's exactly that. It's combining musicians with sacred geometry, with science, with dance, with meditation, and with all the technology. But it's, yeah, exactly that. Getting everyone together so there's all that cross pollination, but B, giving everyone a transformative experience that's, I say, crossing technology, science, and music. So that's gonna be pretty fun. Yeah, maybe we should roll back. So we both met in Orlando, I think, in 2021. And again, it was actually, well, I might as well tell that story. I was sitting there in meditation, and I was like, oh, I'm I was like, because we basically hadn't run any research experiments in outside universities for a few years, like not since we've been in the States. We did some in Europe when we were in Europe. I was like, oh, I bet. I was like, oh, I bet if there's any scientists that are studying this space, I bet they're probably coming to Vent. And I didn't actually know that any research was going on at that point. And then literally, I walked out, went to lunch, sat down to somebody who knew you. We just started talking about research. I think we were introduced probably an hour or two later, and then if I remember this correctly, I think I said to you, it's like, well, these sort of effects that you're saying can happen in meditation, they can also happen just through imprinted water. And maybe I'll let you take over of what you thought or said, so I don't want to put words
Yeah.
in.
I mean, I think we were in baby days then, and I was still had my most of my feet into the science world, and it sounded like snake oil, right? I'm like, yeah, water and frequency, and how do you get that into someone, and what does it really do? And so I was very skeptical, right? And I think that's where we started is I said, fine, send me some samples and we'll play around with it. And like most things in my life, where there's so many things that we're working on that I think we put it on the back burner because I couldn't convince anyone in the lab to try water to see if it would change the behavior of a cell. And then finally we did the experiment, and it was same kind of thing with the meditation work we were doing with Dr. Joe. Is it was like an aha moment, right? Why didn't we do this sooner? And it was an amazing effect that I still scratch my head and can't explain.
If I remember rightly, didn't I think you said it was basically because at the time you were doing a similar experiment for the Joe meditators, where you
Yeah.
were basically wanting to see if you could reduce the viral penetration rate into lung tissue. And I mean, roughly, we basically got the same result, but you didn't. In our cases, no one was having to meditate.
Yeah. I mean, the information that was in that water prevented infectivity in a cell, and it was an immune water that we were trying in this, right? So it was targeted towards this immune resiliency. And to see that it worked the way you would predict was astonishing. I mean, it was a night and day effect. It wasn't like, my mentors always tell you if it's something like this that you have to do statistics to look at, it's
Yeah,
probably not something you want to devote the rest of your life to. But if it's this, where you can just look at it with your eyes and like it's a night and day difference, there's something going on here that we need to understand biologically, then you pour your energy and effort into studying that. And so it was this. I mean, it was a night and day difference between the control and the printed water that we were testing out.
could you explain that experiment in a bit more detail for people? Because so you've
Yeah.
got a culture of lung tissue. You died the virus, etc. But I think it would
Yep.
be good to
Yeah, so
explain that.
we were doing SARS experiments at the time, and so it's a non-infective virus that we created in the lab. So the surface of the virus looks like SARS-CoV-2, and so it behaves just like it in terms of how it engages with the cell and sort of injects its genetic material in once it gets inside. What we did is we encoded the internal material with a red reporter, and so this would be a way for us to track how many cells get infected. The material that gets into the cell doesn't do anything toxic to the cell. It just lights them up red, and so we can easily count them on a fluorescent microscope. And so in a control setting, if you have nothing in the system and you add this pseudovirus with the spike protein onto the lung cells, almost 100% of those cells turn red, and you see this bright field of red when you're under a fluorescent microscope. If you incubate those cells with the water and let them sit in it, I can't remember if it was a half an hour, some time period to acclimate to this new environment and this new information that they're seeing, and then come and challenge that cell with that virus, there's way less red that we see the next day. So what it suggests is that somehow that water has changed the cell so that it's no longer responsive to that pseudovirus. It's not responding and being infected the same way that the control cells were. So it suggests that there's some resiliency that's created there.
So, just for everyone, we basically imprinted information using light and a strong electrostatic field. And chemically, absolutely everything is identical. All it is, well, it's basically minerals and it's water. When I say minerals, it's actually sodium chloride, potassium chloride, and magnesium chloride, and the rest is just really pure filtered water. And I say we sent all of these different types of infoceuticals, but the particular one that we wanted to see, it was well, it was called ET2, that years ago had been designed to basically prevent COVID. And I think what would be interesting for people, because people are probably wondering, like, well, is there a difference between each of these infoceuticals? Because like chemically they're identical. And I know that next set of tests, remembering the machine, it was called a paramagnetic.
Yeah, EPR, yeah. No,
Um,
we ran those samples through that, and there were differences, right? And so it's we were essentially measuring noise in the system. And so an EPR is a really fancy, it's like an $80,000 microwave. And so it's shooting microwave energy through this. And as those microwaves go, you're looking at the spin characteristics on the material. And so it's looking at the minerals and how they're spinning and moving and doing things. As you get this really noisy, jaggedy patterns that as an indicator of what's contained in there. And we noticed that there were unique differences in the different products that we had. So there is potential to suggest that it's not the same information in every bottle that's labeled something different, right? That each product is a unique signature.
it basically say the difference in information had distorted the paramagnetic
Yeah.
qualities. So,
Yeah. So it suggests that ED2 is different than ED3 is different than brain one, heart one, or whatever. All of the different things had a unique signature. They didn't overlap.
so let's move on to, I think you had on your desk, was it Cell Driver?
Yeah, this is ED3. I've been putting it in my water in the morning.
Yeah, good. Yeah. Let's say what have I got on my desk? I've got Heart Driver. That's what I'm putting in my water. So, one of the things, obviously, the company and a lot of the things that we do is all about restoring people's energy. So, one of the things that we wanted to see or to measure is what effect does information that was basically designed to optimize the cell, what effect does it have on basically the energy output of the cell? And then I'll let you take over. I think the first experiment was measuring the density of mitochondria.
Yeah, so as I mentioned, we do a lot of mitochondrial biology in the lab. Um, and I think it was a series of experiments that we did with ED3, both in the infoceutical form, and then we also tried imprinting onto the cells as well. And we saw some interesting insights, right? I think the first data set showed that we actually saw an enhancement of mitochondrial energetics in that system. And so looking at mitochondrial function is really complicated. We've diluted down a couple systems to do this. And what we did on your cells is essentially it's a mini version of this test that we're selling with part of Versaia Discovery called MeScreen. And so that is like a test on steroids. It looks at every aspect of your mitochondrial functionality. But one of the sort of elemented kinds of things we do, and what we did with this ED driver and the cell systems we were looking at, is we can pattern unique cells in a dish, right? And so we were interested in heart cells versus muscle cells, skeletal muscle cells for this instance. And so we incubated these cells with this ED driver, ED3 driver, and then we looked at changes in mitochondrial stress responses. So basically, what you do in this assay, which is measuring oxygen consumption, which is what your mitochondria use to make energy, there is this complex network of proteins that form what's called the electron transport chain. Mitochondria are old bacteria that got incorporated into our cells, and they allowed us to make tons of energy. Mitochondrial energetic production is way more efficient than processes like glycolysis that happen in the cytoplasm. For a few molecules of ATP,
Oh, that's a totally separate
yeah,
pathway.
different pathways, right? They use similar fuels, but
Yeah.
mitochondria really take oxygen as the key molecule in this sort of process. The end product is water, and you move these electrons through this gradient to create this hydrogen differential, and then hydrogen gets pumped back into the mitochondrial matrix, and as that hydrogen gets pumped in, you make a ton of ATP at the end of that. You add this high phosphate group to ADP.
So like carbohydrates, sugars, fats, they're not a fuel for mitochondria.
They are. I mean, you can convert them through certain pathways and get raw material that then feeds into the mitochondrial pathway to utilize it.
But it's
Um,
mostly hydrogen and oxygen.
yeah, I mean, you're basically looking at gradients of hydrogen that's being pumped out, and that's the gradient that's being created. And then oxygen is the final electron acceptor in this pathway as you sort of move down this. So essentially, as you move from complex one to complex three, or complex two to complex three to four, and as you're going along, you're creating this electron transfer that then moves hydrogen ions out of the mitochondrial matrix into this intermembrane space, and it creates this high resiliency gradient, right? So you're pumping less, there's less hydrogen inside than outside. And then at the end of that chain, all of that hydrogen gets pumped back in. And as that pump works, it sort of spins and it makes ATP.
Two questions from that.
Yeah.
There's a in this, there's a lot of, I guess I don't know if it's hype, but obviously a lot of people either using NMN as a precursor to NAD, or people do NAD infusions. Obviously the NADH is, I guess, the hydrogen part, but yeah, what do you feel about that whole NAD thing?
Um,
Is that actually going to make mitochondria more efficient, or is
Right,
it?
so NAD in your mitochondria is a source of efficiency. Complexes use NAD as a electron sort of restorating kind of thing. The problem with NAD, which is a problem with a lot of supplements, is they have to get to the site of action and they have to be absorbed the right way. So the mitochondria have a transport mechanism to get NAD inside the mitochondria. And usually with people who have defects in NAD, their defect is not in the amount of NAD that you have in the mitochondria, it's a defect in this transporter. So you can flood the system with as much NAD as you want, but the thing that's needed to get it into the mitochondria doesn't exist. And so they're going to essentially pee out all that stuff anyways, right? And
What
so
I've noticed with NAD, it's like it does make you feel good, but I'd say probably within a day and a half, you're basically back to normal because it's left your system.
yeah, it's left your system, right? And so one of the things we've been talking about is there a structured way to look at mitochondrial disease, and can you then have a functional and a dynamic assessment that gives you ideas of where to start? And so one of the things we've been starting to play around with, and this comes out of, systems that we've looked at with your infoceuticals, with the imprinters, and then the really sort of far out stuff we're doing with MeScreen, is we can get evidence of where there's a dysfunction. And
I
so for
like
a
how
lot
you
of
said
these,
your me screen is more far out than our infoceuticals.
way more far out, right? It's driven towards this idea of not necessarily diagnosis, but this idea of really looking at a personalized sense of what your mitochondria are doing. And then that gives you some ideas of how to sort of move forward in a different kind of way. One of the things that I think we can assess, and I think that's going to become important in this, is there a mitochondrial structural defect? And if there is, rather than feeding the system supplements, we should work on the lipid sort of reconfiguring aspect of this. So can you rebuild the nuts and bolts structure of your mitochondria first, and then have a layered sort of treatment program where you build the structure back, then you start introducing positive elements that create biogenesis and other things, and then you create the next level of thing, which the supplements may be six, seven processes down the road. It's not where you want to start, right? And so that's what we're starting to play around with.
One of the what's a bit of R&D going on before you have looked at it. It's actually up in Encinitas up the road. So there's a clinic there that has these hydrogen water baths, and they've been getting just I say crazy results, but they've been reversing people with all sorts of, like, probably because it's a podcast, I won't say what they've been reversing, but let's say they've been getting incredible results. And one of the ideas that we've been looking at, and they're just about to do it, is basically we're going to imprint the hydrogen enriched water that they're then laying in the bath. So I guess one I just want to ask you about what you think about hydrogen, because there's people breathing it. You can have it in,
Yep.
you can drink it, you can have it in baths. Whether
Yeah.
you think that's a good thing, and then two, we'll actually get to the original, the original discussion of what you actually found with the infoceuticals and mitochondrial density. So
Yeah. And I mean, I don't know much about hydrogen. I've just started. I was at an A4M conference in December, and that's where my exposure to this was. Every other booth had hydrogen, this, that, and the other. So this is something that everyone's doing and trying. I don't know what to think about it yet, right? I haven't tried it. I haven't really read much about it. I don't know if there's a
there's thousands of I know there's thousands and thousands of studies
yeah.
showing all the well, it's mostly inflammation.
Yeah, so it'd be interesting to see how this moves. I mean, it'd be really interesting to look at those people and see what their pre-post responses are in terms of their energetics and the rest of their biology, you know?
Well, let's just describe what you found with cell driver and mitochondria.
Yeah, so we noticed that there was this heightened response of mitochondria to so I was describing the process, right? So the first test we typically run is a mitochondrial stress test. So you look at a baseline respiration, and then we inhibit the generation of ATP by using a compound that blocks this ATPase. So when you do this, the oxygen flux, which we're measuring in real time, should completely suppress and stop, right? And so if there isn't a suppression and the system is still using oxygen, it suggests that there's an uncoupling somewhere in there and everything is not tight and you get a leak, right? And so you can measure this leak state. The next thing we do in this stress test is we add a chemical that's called an uncoupler. And so it uncouples the entire gradient of hydrogen from the inside to outside, which then causes oxygen to move super fast through the system in its maximal capacity. And so this maximal capacity is a measure of the how efficient your mitochondria are, but it can also be a measure of how many total mitochondria you have. The more mitochondrial mass you have, the more efficiency and the more drive you have through that system. And so what we noticed with the control versus the ED3 infoceutical in this response is there's a dramatic elevation in that max respiration capacity. And so this would suggest that you're creating this reserve and potential that didn't exist before, right? So compared to the control, the ED3 has this huge reserve potential. We haven't directly tested it, so it could be mitobiogenesis, so you're making more mitochondria to create that potential, or it could be that you're making the mitochondria that are already there more efficient.
My suspicion is it's more efficient, but I
There's
say
a
that
couple
just because I
things
was
that would,
there when obviously when Peter, who's dead now, but he was the original researcher who made the cell driver. But it when he was making it, it was all about making the pathways more efficient. I
yeah.
definitely don't remember him thinking he was going to grow more mitochondria.
Right. I mean, this is in a very acute setting, so I would suggest in that experiment we did, it's probably the efficiency aspect, and there's something that's being tweaked. If you were to treat the cells consistently for days, or if someone's taking this for many, many months, there may be a biogenesis component to that, right? So there's certain things that will increase mitochondria, exercise, calorie restriction.
Do you think that also means there's like I mean, it's basically a general protective effect?
Possibly. I mean, we tested this, that effect was in a muscle cell type. It'd be interesting to see if in other cell types it creates the same thing. One of the things that I think people forget about biology is every organ, every cell is uniquely different, right? It creates its own environment. And that's why your neuron is your neuron and your heart cell is your heart cell. Structurally, they look very different. Metabolically, they look very different. And they're going to respond dramatically differently to the same stressors, the same stimuli, the same chemicals. And so it'd be interesting sort of to see if there's a global effect versus not. And I think you're already doing these experiments. And the sad part is we're not collecting any of the data, right? There's people out there that are taking infoceuticals all the time, and we could be collecting samples from them to run these kinds of large studies.
How do we go about that? Because
Let's get some blood on a me screen card and test them
yeah,
through this
we can
process.
definitely do that. We can definitely do that.
I mean, there's other ways to do it, right? I think you can monitor their biometric profiles on Garmin's. A lot of people wear Garmin's or devices, and I think that's where you guys are headed as well with some of the things you're doing with the technology. If there's ways to track activity, sleep, those sorts of things, all of that could be ingested into a large program, and we could start thinking about how imprinting and changing and drinking these infoceuticals and other things are going to change biology. I mean, there's lots of ways to measure this.
Just on the wearable, I mean, one thing we didn't mention. So the experiments, they were both run with an infoceutical, but we also created a mini imprinter, which is basically a sort of like a prototype version of the wearable.
And by and large, I mean, it was very parallel results. Some were more dramatic with the imprinter than with the infoceutical, and that makes sense. And some effects we couldn't reproduce. So I think there is quite a lot of overlap.
Yeah, and just for the audience, so we've been making this wearable that'll detect, correct, and protect your energy and emotions, and in straightforward terms, instead of having to take an infoceutical and drink a glass of water, it'll basically be imprinting into your blood straight away, 24/7, exactly what you need.
Which is that's going to be really exciting to see what comes out of that.
Yeah, especially when we run it with all of the me screens, which
Yeah.
is nearly there.
And it goes, I've been thinking about this. I looked at some of the data yesterday just to prepare for this. And one of the things that was a curiosity, and this goes back to this evolutionary hypothesis that I was talking about. We saw this weird shift in membrane biophysics, right? With this ED3 infoceutical and the driver. We saw this increase in the rigid to fluid ratio, and we saw an increase in this rotational, or we saw a decrease in sort of this idea of a rotational correlation. And so if there's something this goes back to this EPR, so this machine, this fancy microwave that we used to look at the differences in the different chemical structures of the different infoceuticals. You can also label membranes with a specific lipid probe, and you could see if it's hindered in unique ways. And so now there's a metal tag to this, and it basically is a microwave. So things spin, and you could see the hindrance of this spin. And so if something is being hindered, it decreases its rotational correlation, and that then suggests that it's sitting in a membrane that's more rigid than fluid. So if a fluid membrane has less lipids and other things packed in it, this thing spins around really fast and the rotational correlation is really high. If there's a lot of cholesterol and other things packed in, it's so tight that this thing can't spin. And so we're looking at that dynamic. What we noticed with ED3 is that there was this increase in this rigid to fluid ratio. So the membrane was more rigid, and there's a decrease in this rotational correlation. So one of the things I was just talking about is that biology adapted to this high oxygen environment by making more cholesterol. One of the things that will cause rigidity in a membrane is cholesterol. And so this is early, we haven't tested
They
this
think it's
out.
reducing cholesterol.
It's increasing cholesterol, which is, I think, an adaptive thing, right? So the more cholesterol you have, the more oxygen you can potentially bind. And for someone who's like, let's use example as exercise, right? The reason why you stop exercising at a certain point is you're moving into this aerobic to anaerobic phase. You just don't have an oxygen supply that will meet that demand. If your membrane is holding on to more oxygen, then it's delivering this in a tempered fashion. So it takes you way longer to exhaust all of that oxygen because there's so much available in that reservoir pool they've created in the membrane. So that's my sort of straw man out there hypothesis of how this may be changing exercise potential, energetics, oxygen, mitochondria. And we can test this out, right? And so something we're excited to look further into.
Well, if you had to extrapolate, what do you think the benefit is, just to an overall human?
I mean, I would argue probably based on the cell data we're seeing with this high mitochondrial respiration that we see that's when it's uncoupled, and this change we see in the biomembranes with more rigidity, that I would think that exercise capacity would be enhanced, right? You're just handling resources better and you're making more energy and you're making more efficient use of that energy.
Got it. Got it. And one of the other areas that we were looking at was like wound healing. We're particularly interested in wound healing because honestly, because people like to have beautiful skin, etc. And also, actually, there's an awful lot of talk about exosomes, and there's probably a new exosome company pretty much every month popping up as far as I can see. And but not all exosomes are the same. And the thing that we were thinking is like, well, could we get a cell to produce a better exosome? Because honestly, we were thinking that we could license to an exosome company. Of course, the real truth is actually if you just deliver the information to the body, your body cells produce their own better exosomes, and you don't really need to get exogenous exosomes. But yeah, what did you find? Well, actually, let's describe the experiment first, and then say
Yeah,
what.
so what we can do, cells are amazing, right? They're not destroying an animal in any way, they're not harming you in any way, but you can model lots of diseases and processes. So you have a plate of cells, right? It's confluent, meaning that they're growing all over. When you look at it under a microscope, there's no space anywhere. And so people have over the years developed a wounding model for cells. And so you take this essentially a comb and you just scrape into this dense pocket of cells and you create an injury, right? And so now you have a space between these pockets of cells that's completely empty of cells. And so what wounds do is this is basically a tear in your skin, right? Imagine that. And so then what happens when you're healing is cells come in and migrate to that space and they fill up that space and they come back together. So your little sort of open wound on your skin will eventually form a scab and sort of everything comes back together. So in this culture environment, what happens is when you create this middle space, the cells don't like that and they start to sort of invasively grow into that inner space and heal that wound that's existing, right? Because they want to be communicative and interacting with each other. So we can track how quickly they come into that space as a measure of wound healing potential in this in vitro assay. So then the question becomes, how do you track the cells that are moving into this? So what we did is we labeled them with a fluorescent reporter. So there was this green fluorescent reporter. So all the cells light up green, so you can look at them and you can, so when you see the little scratch, it's essentially dark because there's no green in that space. And then you can track this over many, many days to see how quickly those things come together. In this assay, we didn't have to go more than 24 hours because those cells started to invade and grow into that space very quickly. And so what we noticed is that with the ED3 infoceutical, there was a very quick spike of cells moving into that space and closing that wound. And then it sort of trails off and drops, and you see that healing process. The other thing we played around with was we took ED3 and exposed cells to that ED3 and collected off the stuff that comes from those cells, right? And what an exosome is basically for decades, people thought it was cellular garbage, that cells just slough off pieces of their membrane. It has no functionality and wasn't going to do anything. Turns out that when you actually concentrate and look at these things under an electron microscope, they have a shape, they have a size, they have a density, right? And when you look at them in really fine detail, there's things inside them. There's nucleic material, there's mitochondrial DNA, there's proteins, there's metabolites, and there's really unique components to the cell membrane that makes these up as well. It turns out that they're not just arbitrary garbage that cells release. There's actually a packaging process that happens in cells, and they package these exosomes with unique material that then changes biology. I mean, the far out things that people have discovered, there's exosomes that breast cancers will release that then go to your blood-brain barrier. They open the blood-brain barrier up and allow that cancer cell to move into your brain. So one of the primary sites that breast cancer metastasizes to is your brain, and this is an explanation for it. And it's an exosome-driven process, right? These exosomes create that disruption of that barrier, allowing that cancer to migrate through. There's positive elements as well. So there's lots of ideas around youthfulness and people who are young are creating these molecules in their blood, in their platelet-rich plasma, in their exosome profiles and other things that may have resiliency. Stem cells create exosomes, right? So stem cells will package certain material, put it out into the space, and then people think that this is what's creating this paracrine environment that everything's being bathed in that creates resiliency. So that was the idea we had is could we use ED3 to drive this cell to package exosomes in a uniquely different way? And then could we use control water to then look at just normal packaging of material? As we noticed is that there was an early difference between in the same wound healing assay between the exosomes that came from a control cell and the exosomes that came from an ED3 driven cell. There was a lag in the response, but then we saw a separation with ED3 from the control. So it takes a little more time than just adding straight ED3 to the system, but there is this unique effect of ED3 in moving these GFP cells into that space of healing. So early data suggests that
I mean,
maybe
in other words, it increases the speed of wound healing.
increases the speed of wound healing. There's a slight delay in it, but it looks way better than control, right? And so maybe there is a better exosome you can create with ED3 that may have some biological potential.
I mean, well, my thinking is likely you can create better exosomes with every single one. Yes, we have kidney driver, heart driver,
Yeah.
liver driver. You can make better exosomes for all of those organs.
Yeah, and I mean, you can, start thinking about how to sort of look at this in humans as well. And so people who are on infoceuticals, it'd be interesting to do an exosome profile on them and see if they're creating an exosome profile that's creating some of this biological effect that you see, right? At a global level.
So it's pretty. You can just take the blood and see everyone's exosomes.
Yeah, you can take, I mean, we're doing it off of the MeScreen cards as well as a pilot study right now. And so we can take the serum and actually shoot it into our system, which is called a nanocyte, and create an exosome profile. If you take about 10 mils of blood from an actual person and then get their plasma, you can actually extract which is fairly easy to do, right? You're not going to miss 10 mils of blood, it's nothing. You can then purify the exosomes and actually sequence them to see what's inside, which would be amazing to see.
Can you recognize exosomes that have come from different organ tissue?
There is a way. There's
Because
an
we would know
amazing.
we would know all the infoceuticals, that
You could. I
with
mean,
all
there's an
our
amazing guy at Cornell who we worked on a NASA project with, and Chris's lab is phenomenal in terms of sort of machine learning and putting things together and looking at sort of big data. And so I think he's developed an algorithm where you could look at the content of an exosome and determine where it came from.
So before I mean, I'm just curious. So before exosomes were realized that they were in effect a signaling molecule, um, I guess how in normal biology were scientists saying that cells communicated to each other?
I mean, the physical nature was like they're connected, right? Lots of cells are connected to each other and they form this communicative network. There's organs that are visible, clear examples of this. The neurons connect to each other through the synaptic connections and they move information through chemical mediators from one cell to another. And there's electrical communication as well. The heart is the other example. Every cardiac myocyte is connected to the other myocyte, and they form this syncytium, and they couple with electromagnetic coupling, right? So there is this electrochemical gradient that they create, and so every cell learns from the other cell. What's even more interesting and exciting in this is I think one of the primary conduits for this connection is likely mitochondria, right? So mitochondria inside your cells of a heart are also connected to each other and they connect to other cells, right? And so there is this sub level of connection that you see at the heart. Other organs are organized because they communicate with their cells, right? And so livers are livers because there's hepatic cells in them that form this connective information. What exosome sort of solved was how do you communicate from one organ system to the next, right? And so if your heart is experiencing joy, happiness, other things, how does it reflect that joy, happiness, other things to all your other organs to make them resilient? Well, it shoots off these exosomes, it packages these things with genetic material. They get into your blood circulation and your blood takes it everywhere, right? So it goes and circulates. These exosomes, because they have a membrane component to them, we were involved in a paper long ago that actually showed that how these exosomes re-enter a cell. So now you have this exosome with genetic material floating around. Let's say your heart releases this. And this now finds a cancer cell. And let's say your heart's creating some protective molecule that's anti-cancer and it's protecting from cancer biology. That exosome leaves your heart, and then it finds that cancer cell somehow. That's one of the open problems in this and sort of conundrums is there homing effects, right? So is there an exosome that's particularly made and driven to go to a particular site in the body? And does it have a chemical or signature that homes it to there? We don't know.
I've always wondered about that, like how, because you hear about that in the exome world, you hear about it in the stem cell world,
Yeah.
but when blood, I'm just, picturing an exome in your vein, and it's just, just going with the flow. How on earth can something hone rather than just sort of follow the stream of a river, really?
I think it's because of the complement of things that are on the surface, right? So that surface membrane has proteins and other things that are unique. They come from a particular organ, but they're being packaged in a certain way that they'll recognize where they need to go, right?
So they basically flow right throughout the body, but when they get
Yeah,
to
until they recognize where that pattern will sort of hit.
interesting.
And so now it'll identify a target, right? So once it gets to that target, it actually internalizes into that cell in a toxic way, like a virus or bacteria, other things would do. It gets taken up into that cell. And now that cell can use that material to respond in a unique kind of way. The example for cancer would be if this is a therapeutic, then, right? That therapy is being delivered in this exosome. It gets internalized into that cancer, and whatever that material is, whether it's the protein or the nucleic acid, the DNA, RNA, whatever's packaged in there, is then going to change and transform that cancer cell to behave in a different way.
Have you ever looked at like biophoton cellular communication and like other ways that, well, like how cells can communicate through fields?
We haven't. I mean, that's a conversation that a lot of people have brought up recently, and my mind hasn't captured how to do that yet. But it'd be interesting to see, right?
I remember one experiment I read years ago. I think it was done about 34 years ago. I think he was called Gershwich. But he was looking, well, he basically had, I think, two, a group of cells divided by a sheet of glass. But when you put the glass there, because ultraviolet wouldn't travel through the glass,
Yeah.
basically the other cells wouldn't react. You take the glass away, and they would, the basically meaning that there was communication going on through an ultraviolet photon. And then there was a lot of work done. I think he was working on it for about 30 years, which was Professor Popp in Germany. Um, yeah, and then obviously from our Peter Fraser point of view, who was the co-founder of NES, who's mapping out the body field. He well when from his point of view, he saw that there were, like, well, he called it the human quantum electrodynamic field, and that got condensed to the human body field. But basically, that's following that action at a distance idea that every single cell is interconnected. From his point of view, I think you have like 3 trillion operations happening every single second. You've got
Yeah.
50 trillion cells, and it's like, well, how on earth can all of that be coordinated in this perfect harmony if it's just through chemical messaging? And like that would basically be,
I mean, I think there's some truth to all of that, right? I mean, people have described matter as frozen light, right? I mean,
yeah.
we're essentially frozen light.
Yeah, well, I guess that's what we need is this campus of musicians, physicists, and
I know, get everyone into a room together and just sit and drink tea and coffee all day and answer the problems of the world.
well, we'll get them all into Zenergy. And I guess on that note, so Professor Patel, he'll also be speaking at Zenergy in Denver on the 3rd to the 5th, and obviously he'll be going into detail on some of these topics. Actually, yeah, we should probably perhaps say what some of the research that we're going to be continuing to do. And
Yeah. I mean, I think one of the hot areas in this is the energy piece, right? How is energy being impacted by these drivers? Can the imprinting do the same thing? Does it impact every cell the same way? I think we've focused on healthy cells to this point. I think we're going to turn our attention to disease processes as well and see if this is a way to fix cancer, if it's a way to fix diabetes. It's a way to fix things that have this crazy abnormal energy balance kinds of things. And so I think that's a positive end. From a biological perspective, we're always very reductionist in how we sort of look at things. And so I think the membrane could be a big answer to what these things are actually doing. Most people sort of ignore the membrane when they think of the most important part of the cell. And my argument would be it is the absolute most important part of the cell because the cell doesn't exist unless you have a membrane. There's lots of data and studies out there that suggest that you could put a cell in an electromagnetic field, pulse that field in a unique kind of way, and that cell will become a heart cell or a neuron cell depending on that frequency that it's being emitted and sitting in. What this suggests is that membrane becomes the initial sort of capture of that information in the exogenous world. And so I think looking more at how the membrane behaves in a living system could give us a lot of answers about how we engage and interact with frequency and information that is invisible, right? And so I think that's an area. The other big thing we've talked about, and I think we finally should sort of launch a project around this, is how is this changing the molecular biology of cells? I think this is where you start to cultivate relationships with traditional cell biologists and biologists who have their mind completely focused on the genetic material that's being manipulated in biology and life. And so I think having answers around, is there a genetic signature that's being manipulated as well, could be a very fruitful avenue of investigation. We've talked about a series of experiments around that as well.
Perfect. Is there any site or anywhere that you would like to point people to? I mean, I know you mentioned your MeScreen test, but yeah, is there anything you would like to point people
Yeah, I'll
to?
send you a couple sites, the MeScreen site, our UCSD site, and then if you want to meet us, we'll be at this conference in October, but we're also at a lot of Joe Dispenza events doing all kinds of crazy research where we're looking at how individuals couple in a social system and in communities and things like that at a very biological level. I'll put a plug in for a big study we're doing in Denver over the summer. We're looking at paired individuals, and so we're going to be looking at young and aged pairs, and we're trying to see if there's something of a transference of systems in biology when you pair a young person up with an older person. And we're looking for 3-decade differences. So if you are interested and want to come out there and enjoy a week long at an amazing event that'll change your life, that would be an amazing thing for you to sort of engage in.
Perfect. Well, yeah, thank you very, very much, Hamel, and well, I've no doubt you'll be back on the podcast soon.
All right, it was good.
Thank you. If you want to learn more about bioenergetics, including the transcripts and downloads for this episode, please go to www.bioenergetics.com. If you'd like to do one of our free introductory courses on bioenergetics, then please go to bioenergetics.com/intro. Or if you'd like to do our free intro course on GIST, then go to the gistprocess.com/intro. If you'd like to experience our voice scan, see a practitioner, become a practitioner, or a bioenergetic health coach, then please go to www.neshealth.com, where you'll receive our free ebook, Restoring Your Health with Bioenergetics, along with our movies.